Thank you so
Green
much
light.
once again. And for MBC, really appreciate your support and all the PerkinElmer team support. So the screen is yours.
Thank you very much, Flavia. Many thanks to you and all the efforts that you put in. I know that we have asked a lot of you, and you've been very communicative and responsive. It's been great working with you. I know shRNA, myself, and all the rest of the people on the team are really looking forward to continuing this relationship. Thanks everybody else from the Horizon internal team for coming on. I think this, either way, will be a good summary of some of the things, some of the efforts that we're trying to tackle within Horizon and the way that we think about engaging with our customers. And thank you for everybody from MBC Biolabs that's come on. Me personally, I love working with startups because it makes me feel like we have boots on the ground, and the conversations that we are able to have with you are so meaningful and impactful that it just excites me every single day. To that end, this talk, Enabling Your Success During Startup and Beyond, it's not a very technical talk. It's more of a conceptual exploration about how Horizon can help you acquire the data that's going to be most critical for you at the early stages of startup, with your ultimate goals being progressing your compounds to a state to where you can get additional funding. This session is pretty brief. It's pretty light and breezy. It's only about 15 minutes. We will be having a Q&A session after this at each of the sites. Eleni will be at the 953 Indiana site. She will be in Tar Heel blue sweater. I think she also has Tar Heel blue painted nails, so you probably can't miss her. shRNA, if you can just type in your a way to distinguish you into the chat box so people can see you. But they will be in the lobbies standing next to hopefully hot and fresh pizzas if the timing all works out for the deliveries. Hopefully what will happen at the end of the session is you will be interested enough, because this is just a real introductory session, to reach out to us after this session and have a conversation about why Horizon would be a good partner for you in your steps moving forward. Okay. Horizon has worked with a lot of small biotechs over its lifetime. We've also worked with a lot of large pharma, but routinely we work with biotechs. That's our core group. And we know that biotechs face a challenging path to success. They have a lot of pressures that are put on them by the market and by their board and their investors. So making a decision on which data is most important to you is extremely important, especially at a point in time whenever you have very little precious time and very little resources to do these things. So the types of questions that Horizon can help you answer, and you can start to evaluate in the broad scheme of the bigger picture of things, are what tissue types or what indications would be best for you guys to play in. So what market is going to be the best strategic option for you? How to assess dosing, or what dose to use for your compound within that indication that you end up choosing. And then most importantly, but is generally a little bit farther down the line, is understanding at least a little bit of, or comprehensively understanding your the MOA of your compound. So the way that we can help you answer these questions is threefold. We can test compounds in a collection of cell lines. And this allows you to identify genetic or cellular dependencies to your therapy. Or we can generate the ideal model for you, which allows you to have a system to run functional studies, which is extremely critical in moving your data forward whenever you need the depth of understanding for that next round of funding or for those next conversations with the board to justify resource allocation. And then finally, we can screen for genetic interactions. And this is critical because this can potentially identify novel biomarkers that can be used in the clinic or that can be used as you're working through a lot of your R&D. A recent report out of the Cancer Medicine Journal actually showed that breast melanoma and non-small cell lung cancers that had biomarkers associated with them were 12, 8, and 7-fold respectively more likely, or had more likely to have to be approved within the clinic. They had those fold higher probabilities of success in the clinic. So the way that we can test your compounds is by utilizing cell panel screening. The way that we can create the ideal model is through cell line engineering, and the way that we can evaluate genetic interactions is through functional genomic screening. These different services that we offer are represented within a flow that it looks like it cascades from cell panel screening to functional genomic screening. But in reality, each of these phases can be introduced or can be entered into really at any point in time and at any phase within this workflow. The reason why we have put cell panel screening at the beginning of this flow is what we've noticed is that this is a really cost-effective option to get a lot of data and a lot of different data, and also allows you to orient yourself towards market, which we feel like is one of the more critical questions that a startup needs to answer at the beginning stages. But it can also allow you to answer peripheral questions such as the dosing that's required or if you need to move into combination therapies. Functional genomic screening is pushed to the end because it's generally a larger investment. That being said, the growing philosophy out there in the market is that there's at least a requirement for minimal MOA studies to be performed on compounds. There's data out there that shows that if you lack MOA data, the potential consequences are you can have incorrect biomarker association or incorrect biomarker identification, which can lead to a whole cascade of effects all the way through to the clinic and patient recruitment. Or you can have interlab variability, which is extremely important whenever you're thinking about obviously moving your compound into the clinic or trying to get that asset acquired by a pharmaceutical company that is going to want to have that asset be translatable. As such, because of those factors, compounds that have MOA data associated with them have a far higher likelihood of being approved within the clinic. And also it conforms and meets the expectations that pharmaceutical companies have that assets that they bring on board have MOA data associated with them. So as a result of that, MOA is nice because it doesn't really matter what your goal is. If your goal is to push a compound all the way through clinic, or if your goal is to just push it through discovery and then sell it off somewhere else, or if your goal is to just sell the asset or be acquired by a pharmaceutical company, there's going to be, having MOA data will support all of those goals and help you achieve those goals. So let's work through an example of how a company could work through this flow. So let's start with Startup A, who has 2 years of funding for a compound that shows efficacy within gastric cancers and epithelial cells. They decided to run a cell panel screen with us or with anybody in general. It could be any CRO in this case. and they choose a large swath of cell lines that come from different cellular backgrounds and span different indications. What was shown within this study is that their compound was most effective against cells that had point mutations within a particular subfamily of protein receptors. And that sensitivity was particularly enhanced within epithelial cells and within gastric cancers. And they also showed nominal effects on liver and kidney cancer cell lines. To evaluate this particular protein receptor, surface receptor family a little bit further, they use cell line engineering services in order to generate a series of cell lines that had different proteins within the signaling cascade expressing GFP, and then also accompany these with SNPs within the pathway as well to understand how these things modulate the response to the compound. In addition to that, they adjusted the moieties on the compound in order to create a few different leads, and then they used this system to evaluate the uptake of that compound and the effect that it had on the GFP expression and the signaling within that pathway. So from there, they now have an optimized model that they can throw into functional genomic screening. In this case, it was a whole genome CRISPR screen that was run. And this was run within a gastric cancer cell line, a kidney cancer cell line, and a liver cancer cell line. And the result of this screen was that they were able to identify potential genes that could be utilized for synthetic lethality within the clinic. And they also identified indications that might be better to be supported by a combination therapy approach rather than a single agent therapy approach, because the cell panel screens only showed minimal effects, whereas there were increased and heightened effects within those indications whenever they were accompanied by a whole CRISPR or genetic knockout. And the net effect of this entire story, the net effect of this entire journey is that at the end of working through a CRO in order to get all this data where it was all integrated, they were allowed, they were able to pursue a previously unaddressed market within this compound class. And then that allowed them to sell off this asset to a large pharma. So partnering with a CRO that has integrated discovery services, such as the CRO in this situation, which just happens to be us, but it could be other CROs as well. But partnering with a CRO that has that type of portfolio allows for seamless transitions during project phases. Which is always great. We can always coordinate things just to make sure that logistics are easier. It allows for retention of project knowledge, especially the knowledge of your compound specifically, and allows for those pieces of information to be used to make biologically relevant decisions. It also strengthens communication through all the relationships that are just built throughout that entire journey. And I would argue that that's probably the biggest reason why you'd want to go with an integrated solution like this, because the relationships that are formed within those and the trust that you have in the people that you work with means everything in those situations. Because at the end of the day, what's most important is for you to get data that you trust. And in order to trust that data, you have to trust the people that are running those screens for you, and that they're communicating things to you truthfully and effectively throughout that process. What can be done with the data that is generated from the services that are provided, like by an integrated service provider, is that can provide you clear direction in what indications or what market you want to play in, as mentioned earlier. And it can also allow you to have these conversations early and quickly, so that you're not wasting time and money, because you want to make sure that whenever you make a decision, you don't go down a rabbit hole that doesn't lead anywhere. You want to make sure that you've made the right decision and sussed it out on the front end. And that's something that we can help with. So we're coming to the end now, and I just want to wrap it up and comment on a few different kind of nuances or unique and subtle benefits of some of these screens that Horizon in particular runs. Cell panel screening in particular is nice because it allows you to quickly understand and have strategic conversations around the ideal market for you. So you're essentially having strategic conversations at the very front end of your stream instead of midstream and having to backtrack after you've already invested a lot into the compound at that time. Cell line engineering, if you work with Horizon Cell line engineering in particular, we have an automated cell line engineering system that just makes cell line engineering a breeze. And it's always nice to rely on somebody that has that level of infrastructure because you can trust that you're going to get it right the first time. And that allows you to focus your time on things that are maybe more in your wheelhouse if you're not necessarily like very comfortable with molecular engineering. And then finally, and I think most importantly, is functional genomic screening can help you start to meet raise up to the expectations within the market that there needs to be increased stringency and an increased level of MOA characterization for compounds that are coming to market and moving through clinic. And running functional genomic screens also allows you to gain credibility at the next funding meetings that you have because it's starting to gain recognition within the fields as being a pretty essential part of the discovery journey. So I'd like to thank, like I said, that was quick and breezy. I'd like to thank everybody for taking the time to listen, and we can field some questions on the call. I'm always happy to do that. There should be pizza out in each of the lobbies now if you're feeling hungry, and we can just have the Q&A sessions in each of the lobby spaces.
I'd like to thank you, Jeff, Eleni, and Serena for being on site and supporting our community. We will also share the recorded version of the seminar with all the community, and hope everybody enjoys the pizza. Well, if nobody else has any questions, I will stop recording the video, and I would like to thank you all, PerkinElmer and all the attendees here for the seminar, for the webinar, and we are looking forward for the next one.
Awesome.
Thank you very much, Flavia.
Thank you, Serena. Thank you, James. Thank you, Jure.
Thanks, everyone.